Myself, Sunil Gairola, I look after the quality control department at Serum, and I joined Serum in 1994, and since then I had worked in various faculties of, I would say, quality control of the products, starting from DPT to virology to the recombinant products. And prior joining Serum, I was a part of a national control lab which releases the product for distribution in the domestic as well as export market. So my academic background is I am a microbiologist by training. I did my PhD in Pertussis, and I was there in the national control lab for almost 12 to 13 years, and I did my PhD from Himachal Pradesh University, which is nearby Kasauli. Kasauli is a hill station which is around 6,600 feet above the sea level. So what today we will be discussing, it is a sort of an interactive session. We are not very much trained for delivering the lectures. So what we can do, we can have good interaction, and all three of us will try to answer your queries if they are. So we will cover these 4 topics. One is the vaccine starting from the Pasteur era till the genome, and second is on the adjuvant discovery, and third we will do a case study on the Menafrivac. It is a meningitis vaccine which is developed by Serum Institute in collaboration with PATH and WHO, and it has a very good case study which has almost I would say we have distributed around more than 250 million doses, and since then, after introduction of the product, there are not even a single case in the immunized cases. And then we will also touch upon the recent biological scenario in terms of the biosimilars and monoclonal antibodies. So if you want, you can come a little ahead so that we can interact more closely. So we start with the word vaccine. The word vaccine started from a term called vacca. Vacca is a cow, which is termed in Greek. That means the first vaccination started from a relation which is called cow and a vaccine, which I will discuss at a later stage, not just now. And vaccine are developed as a part of the product which are antigens. And what are those antigens? We term it as the attenuated or killed microorganisms or proteins derived from them administered for prevention, treatment or amelioration of the infectious diseases. Now, once you inject a foreign material inside a body which may be which is a biological material in terms of protein, polysaccharide or other metabolites, the body recognizes it as a non-self, and then they try to do a defense against it. So that is the first definition of a vaccine. Now if you go to the Wikipedia, it has given a little more elaborative definition of the vaccine. Vaccine is a biological preparation that improves immunity to a particular disease. Immunity term we will again discuss and in the later part of the slide. A vaccine typically contains an agent that resembles a disease causing microorganism and is often made from a weakened or killed form of the microbe. The term weakened means you just delete its pathogenicity. You maintain the antigenic part of the product so that the antibodies are induced against those antigenic fragments, and you lower down the pathogenicity. The pathogenicity is lowered down by different procedures like attenuation, killing, okay, or by removing certain genes. And these are the agents which stimulates the body immune system to recognize the agent as foreign, destroy it and remember it so that the immune system can more easily recognize and destroy any of these microorganisms that it later encounters. An ideal vaccine is the one which produces the same immune protection and usually follows the natural infection but without causing disease. This is what I told. I mean, it's the same organism. So what we did, attenuation is a process where you passage the organism from one to another till the virulent markers, they are suppressed, and only the antigenic fragments which are protective, they remain intact. and it should generate a long lasting immunity. It should not happen that you immunize once and then after some time you lose the antibody titer in the body. There is an immune mechanism how the antigen can express the memory cells, and then these memory cells, they in future recognize that antigen and they react more fast as compared to the previous. So basically the term prophylactic is not therapeutic like pharmaceutical product. The prophylactic has come from the word vaccine because you give it as a prophylaxis. You prevent, you just predict that by introducing these antigens you are going to prevent the future infection if it is caused by the same type of an organism when it comes into the life of an individual. And it of course it needs to prevent the spreading of the infection in the human body. Now coming to vaccine is directly linked to the immunity. Now coming to the immunity, there are two types of immunity, what's called innate and adaptive immunity. Now innate immunity doesn't have a immunological memory. There are non-specific barriers we have like skin barriers, then the secretions of mucosa is a saliva, then the lacrimal secretion in the eyes. So these are the non-specific barriers. You have a nasal mucosal which kills or inhibits or restricts the organism to penetrate. Once it penetrates inside the body, then these non-specific barriers, they go away, and then comes the immune system in picture. And for adaptive immunity, you need to have the immunological memory. The foreign antigen is recognized as a specific manner and immune system acquires its memory of it. How it attains the memory we will discuss in the subsequent slides. I am purposely going slow. If you have any question, you can interrupt in between, because maybe I think this is something which is known to you or not known to you, I am not very sure. So if you have any small questions or even larger questions, you can ask. Three of us will try to answer it. Okay. Now, once the antigen penetrate into the body, it is captured by the macrophages, and these macrophages take them to the antigen presenting cells, APC system, or antigen presenting cells to the MHC part 1 or MHC part 2 systems, which gives the direction of production of the either B cells or T cells. Okay, there are two types of lymphatic cells, either B or T cells. There is a immediate importance of the T helper cells. Now these T helper cells are the one which are very critical for the memory. I have made a good sort of a schematic diagrammatic picture where you will see how these macrophages and these T helper cells, they interact, liberate cytotoxic killer cells as well as T helper or T suppressor cells. And on the other hand, the T cells are basically derived from the thymus. Thymus is a gland which is just in front of the heart, and in the sternum it has two lobes. So through the lymphatic system it produces the T cells, and the B cells are basically from the bone marrow. They started from the bone marrow, but they are available in part of spleen as well as the lymph nodes. And there are 5 types of antibodies, which are IgG, IgM, IgA, IgD and IgE. And IgM are the one which are first formed in the body. So if you want to detect the recent infection, you look for the IgM antibodies. And IgM antibodies are the one which are very strong. They are the pentamers, and these pentamers are linked to each other with the disulfide bridges. The disulfide bridges are intra as well as interdisulfide. Now the antibodies are functional as well as non-functional. We say functional, the one which has the neutralizing capacities, because once the antigen comes and these functional antibodies, they bind, it's a covalent bond. It doesn't go away. Okay. And there are non-functional where it is not completely neutralizing, but they just mask the antigen but keep the antigen exposed, so infection can still happen. So the neutralizing antibodies are the one which are functional IgG type antibodies or IgG subtypes. Now there is another term called opsonization. Now opsonin is a sort of an antibody. The word opson came from a Greek word which is a side dish. Side dish means you need to eat. So the opsonization by the phagocytes is a phenomena where you take out the organism and they engulf it. So these opsonin antibodies which are there in the plasma, they coat on the surface of the infecting agent, and these phagocytes, they recognize it and engulf it. It can be by dendritic cells, it can be by macrophages, it can be by the phagocytes. Now there is another component in the body, in the plasma, which is called complement. It's a heat labile, and there is a complete complement cascade. Now this complement, it is activated by two ways. One by the alternate pathway, another is a classical pathway. Now in both the ways it forms, one is called C3 convertase, and then it forms a cascade where 8, 9, 10 complement fractions are attached, and this once 8, 9, 10, 7, 8, 9 this complement cascade is formed, what it does, when the antigen and antibodies, they interact. For example, there is a pathogen and there is an antibody circulating due to the immune system. It attaches to the pathogen, then complement gets activated, and the C portion, FC portion of the complement attaches to the cells and causes lysis. So it's called complement dependent lysis. So you can see through this that how much defense mechanism we have within our body, starting from non-specific to a specific memory to certain components which are present in the body, but still people suffer. Now broadly, to describe immunity, I have just created this table. The immunities are of two types. One is a naturally acquired, another is artificially acquired, besides the non-specific. Now naturally acquired is when somebody gets an infection through some pathogens, the antibodies are formed inside the body, and again naturally acquired when the antibodies, they are passed through the placenta to the fetus. So this is an indirect. Now if you see the second part and see the current scenario of India, the India is now completely declared called a complete eradication of a neonatal tetanus. Neonatal tetanus was one of the major concern, and a lot of infants died because of the neonatal tetanus, and it is only because of the immunization of a tetanus toxoid component to all the pregnant ladies in the first trimester or in the second, even in the second trimester. So the antibodies which are formed through the plasma, through the placental, they protect the fetus. Even in case of measles, now the measles vaccine in India is given at an age of 9 to 12 months. But if you go abroad, the immunization is maybe a little early. People, they try to give it at 6 months of age. And this difference is because of the maternal antibodies which are there in the fetus. So once the child is born, till 6 months, the antibodies against the measles are present. So if you try to vaccinate the child at that age, then the vaccines become little ineffective because you have already the antibodies present against. So this is all research has been done on it. So how the scheduling of vaccination is done is not merely that you select a 9-month. So selection of dosing and immunization schedule is based on all the data which is available. Okay, and for a human being, you need not to introduce a pathogen to get a immunity. So there we do what? We create, we mimic something which is just like a pathogen, and these are the vaccines. And then you acquired an immunity by injecting the vaccine, and artificially what you do. For example, there I'll give you example of 2 cases. One is the snake bite. Now venom is a very good antigen. Okay, but what it does? It has a neuroparalysis, it has a hemolysis. It will induce antibodies, but the time is too short and the patient dies. So what we should do with the patient? So what you do, if the body is not able to produce the antibodies in such a short span time, you make ready-made antibodies outside the system. So either you raise in humans, there are cases where you raise in humans, or you raise in the animals. Horses, mules, then sheep, goat, these are the animals which have been selected. So what you do, you give graded doses of venom of all the poisonous type. There are 4 basically poisonous snakes which are there. So against those 4 you have to raise and make either a monovalent or a polyvalent. Why you make a monovalent and polyvalent? Monovalent is very effective once you identify the snake. But in case you are not able to identify, then in such circumstances you have a doubt, so you have to give a polyvalent. It is bad that you are unnecessarily giving 3 more antibodies. If great has bitten, so you are unnecessarily giving the antibodies against cobra, Russell or Ichthys. But under doubt you have to give that product. Now once you give this, the patient is immediately starting responding to it. The venom gets neutralized within the system. So you protect that human being. So this is called the anti-sera, the anti-serum which is raised against the infectious diseases. So we have the anti-sera against tetanus, anti-sera against diphtheria, anti-sera against the anti-snake venom serum. Okay? The gas gangrene, you cannot. You have to immediately give Clostridium welchi. You have to. It's basically an amputation which happens in the gangrene case. So these pathogens have once they enter, based on their pathogenesis, has a very severe impact on the human system. And all scientists, they are working towards how we can save the human life. Now this is what I discussed, the barrier. Once the antigen invades, it breaks the the skin barrier, enters into the and then it enters into the system, and then the adaptive defense starts. From this side you will see the B cell response, where the B lymphocytes they activate, and here from this side you will see the T. Both are thymus dependent, and this is the either from the spleen or from bone marrow. And when antigen enters, it is captured by the macrophages, presented to the APC or antigen presenting cells, liberates cytokines, and then there are native T cells which are there circulating inside the body, which are activated, and then it gives rise to the memory T helper cells, or also activate some suppressor cells. The balance of helper and a suppressor cells triggers a sort of a tolerance. How much should be the helper cell and how much should be the suppressor cell? That balance needs to be maintained within the body. Now you will see that these T helper cells, through the CMI, this is called cellular mediated immunity, leads to the production of the cytotoxic T cells. Now there are 2 types of cytotoxic cells. One is which are naturally circulating inside the body. This is called NK cells, and another CT cells are activated by the cytokines. So these are the one which are by cytokines as well as by the T helper cells. And then T helper cells also, they attach to the B cells, and this creates or develop a memory. Now once they enter, the immediate action is the B cells start multiplying. Okay. Now some part of the B cells, they go to the infecting agent, attack it. Some part goes in the background and create a memory. Okay. And majority of them, they remain circulating. So the moment they put a rush, there is a lot of death of the B cells also. They die within very short time, maybe 4 to 5 days they die also. But the generation is very strong. So they go on multiplying, and the T helper cells, what they do, they tell the B cells not to die, they make them strong by helping it. Okay, the B cells are more stronger once the T helper cells are there. So this is the whole phenomena of removing a pathogen from the body and also memorizing that pathogen once it comes to you in a future life. So here is a concept of vaccine. Vaccine you are not giving to the individuals who are suffering from a disease. Normally vaccines you give to the healthy individuals. And if you see the Serum motto, it is a protection from childhood. So we make majority of the pediatric vaccines. So which are used in the expanded program of immunization. So like BCG we make pentavalent vaccine or DPT vaccine, then there is a tetanus, there is a measles group of vaccine, then there is a polio also. And again polio is of two types, one is oral polio, one is the inactivated polio. Now this oral polio is from a trivalent stage is coming to bivalent because there is a complete eradication of, I would say, eradication of a wild type 2 virus. So that's why now from trivalent it has come to a bivalent, 1 and 3. Because there are certain cases which have been reported which are vaccine related cases. The virus, because it is an attenuated virus, so in certain individuals because of their defense mechanism, because the moment you take an oral polio you go on shedding those in the stool and they are in circulation. So from this type 2 we got certain cases which are vaccine related. So WHO thought to completely go away from type 2 and is supported by dosing of the inactivated polio, so that there is no recurrence of type 2 also. So ultimately maybe they may shift from the complete trivalent 1 and 3 also to a complete inactivated polio vaccine. And fortunately we are the one who has acquired one of the companies in Netherlands which is basically involved in production of the inactivated polio vaccines. Okay. So the Serum philosophy is normally to go for a product where it is most required by the humanity. So even though it's a business strategy, whether we have this high level of profit or a low level of profit, but if it is required for humanity, the management and the chairman, they always decide to, even there is a low profit, still we have to go for it. So this is sort of a philosophy of Serum. So till date I would say that more than 700 million children they have been saved by the vaccination over the globe, and this era of 2011 to 2020 has been considered as a vaccine, a decade of vaccine, and we presume that it will at least save 25 million more deaths and coming down to 5 deaths, saving 5 deaths per minute. So this will be by the products which are in pipeline and by the products which are being developed. Now this is the painting of Edward Jenner on this side, and you will see a lot of people suffering from the pox, and this is the father of vaccinology, Louis Pasteur. So you will see these pictures in a lot of immunology books, but if you see closely you will see the cows over here. Okay. Now this is where I said the vacca has come from. Now this figure not only tells about but it also relates to 3 things. One is they observed that the milkmaids who were milking the cows, they have got the pox in their fingers, and it was observed that even though the rest of the population suffered from smallpox, they were protected. So it's a first incidence of called a cross protective antigen, that there is some antigens who are related to not directly a smallpox, but there is a cowpox antigen which is related to smallpox, and it has given that protection. Secondly, if those are not getting the infection, it gives an incidence of the memory. That means by the cowpox, once the individual is recovered, it has a very strong memory. And when you passage this from one to another, it gives same type of pox, is an indication of the specificity of the organism. So you have a specificity, you have the cross reactivity, and you have the memory. So please remember whenever you come across this painting. Now here you can see the lot of people suffering from the pox, And this was first incidence which happened in one of the remote area in Spain, but then the vaccination of was done to rest of the European country. How they transported that antigen, because there was no cold chain, the virus was, of course, it will die after some time, and then there were no the flights available, so only mode which was there was the ships, the boats. And so what he did, he selected some populations, orphan population, not the population of this orphan population. He took them in the boat and he transferred that pox to one or two individuals, and it took almost 28 days to travel from Spain to another. So virus has to be cultivated. So what they did, they have not injected, they have asked them to share their shoulders. So it is passage from one shoulder to another, and that's how the virus was taken from one area to the other, and the vaccination was done. Now Dr. Jadhav has talked about the GMP and CGMP, but if you see this is a rabbit which has got the rabies. It was opened by a Pasteur, picked up his spinal cord and brain, made a tissue, inactivated. He inactivated by formalizing it and injected, which saved the life. So these are the earlier thought process and the inventions which has led to the discovery. Now this is a scenario which tells about the vaccine development. In early 90s, there were only 3 vaccines which were developed in 150 years. Then in another I would say another 50 years, 65 years, there were only 16 vaccines which were developed, and between 1990 and 2000 there are 10 vaccines which are developed in 10 years. So you will see that the number of vaccines which are increasing is from 3 in 150 years to now 10 in 10 years, and now subsequently you will see many vaccines even coming in 1 year. Now with the vaccination, I will give you an example of 2 cases, 2 things. One is this 2005, and where the South America, whole of the South America is now declared free from measles and rubella. And Dr. Poonawala and Dr. Jadhav both were recognized by the Sabin institution, and they were given the philanthropy award also. So this is because of the vaccination of the MR vaccine in the whole of South America. We could delete it, we could completely eradicate. But unfortunately, if you see the global map, you will find that India has so many red zones where there is a lot of measles happening. Why an Indian company which can eradicate those 2 from the South America cannot do it in India? It can, but again it comes to the vaccine policies, the immunization campaigns, immunization programs, or certain cases where people do not take vaccine only. Vaccine, if you see, is not merely the antigens which are present over there, but then there are so many other things which makes it as a perfect and a vaccine which is effective. It has the adjuvants for certain products which are adsorbed on the surface of antigen, and which helps in giving a much more long lasting immune response. There is a slow release of the antigen once you inject. There is a slow release of the antigen from the body, and the immune system is in touch with those antigens for a longer time, and it is frequently sensitized and gives more sort of an immune response with respect to T or B cells depending upon the type of antigen it is. You need to add certain stabilizers in the product, because these are very labile. Some are heat labile, some are labile. For example, polio, if you take, polio is killed almost to 100% in 4 days if it is exposed at a room temperature. So those product has to be transported under frozen conditions. In addition, you add those stabilizers. Plus, for a multi-dose product, there is a policy by WHO which is called the MDVP, a multi-dose vaccine policy, because once you have, for example, 10 doses or 20 doses in a vial, you withdraw, the dose is withdrawn in the field. So once you withdraw a dose or a second dose, there is a likelihood that you contaminate the vial with some of the organisms. And if you do not have a preservative, these preservatives are the one which makes the organism static, or sometimes they are cidal. They will inhibit the multiplication of the bacteria within the product. So especially WHO has recommended that we need to add preservative in a multi-dose container, not a single dose container, because single dose container you just open it, withdraw and inject. Now this I have already discussed from the Pasteur era, that the basic philosophy was to isolate, inactivate and inject. So I will skip this slide. Now based on the empirical approach, which is the isolation, then multiplication and then injection, inactivation and injection, these are various types of the vaccines which are available, which range from a whole kill vaccine to live attenuated and to toxides. Toxides are what? These are the toxins which are converted to toxide, from a toxic to toxide. Toxides are not virulent, or they are not harmful to the body. So that moiety is removed by, moiety is formalized, I would say, by addition of formalin. And then there are polysaccharide vaccines and there are subunit vaccines. So I will describe the flow chart of the two of the products which we make in Serum. Now based on the empirical sort of an approach, these are the products which are available in various categories, and these categories are live attenuated, killed organisms, toxide, polysaccharides and glycoconjugates. So these are the vaccines which are available based on the empirical approach as on today also. Now this is a flow chart of the manufacturing of a diphtheria. So just I will not go one by one step. What I will do is, first of all, you select a strain of diphtheria which is a vaccine strain. Normally these vaccine strains are given to the industries by the WHO. WHO recognizes those strains and they give it, they distribute it. Earlier it was distributed to the national control or the public health, and then these public health laboratories, they used to give it to private manufacturers also. Maybe we got it from RIVM, the Rickson Institute, which was there in Netherlands. Some strain we got it from Central Research Institute, which is a pioneer in the vaccination, and maybe some technology we also got it from the Hafkin and RIVM. So for diphtheria there is a strain called CN2000 strain. So this diphtheria strain is first grown on a Loeffler's medium. It's a fastidious organism. It cannot grow on a simple medium. So you make Loeffler's serum slope. Serum is used as a source of nutrition. You passage it. From this passage you put it into the pre-seed flask, which has again the papain digest meat medium. It was earlier it's a papain digest meat medium, which is a historic. Now we have converted this meat medium to a synthetic medium, because now the industry is moving from a meat free to a synthetic medium, because meat components are always criticized because of their involvement in many of the factors which are criticized. So you make a small seed flask first, grow that culture on a seed flask, and from the seed you inoculate the fermenter. Okay. Now these fermenters are the vessels where you have your pre-sterilized media. Seed has certain sort of an inoculum with respect to the opacity. You calculate how much, because based on the experience only you do it. What should be the ratio of the culture of a seed to the final volume of the fermenter, you have to decide on experience, plus it is based on the product which you have already clinically proven. You cannot deviate from the manufacturing process. That's where your phenomena of consistency is involved. So prior giving a license, you have to do some consistency, and immediately post license, before marketing of a product, you have to demonstrate the consistency of product. And all those parameters are validated for seed inoculation, for fermentation, and then harvesting, and you process your product according to those validated procedures. Now once the bacteria has grown in fermenters, it liberates the toxin, which is diphtheria toxin. You are interested in the fluid which has a toxin, so you filter it out by using depth filters. Some people use even filtration technology. There are different processes of sedimenting out the bacterial mass and taking out that fluid media which has a toxin. Now this toxin and the medium is termed as a crude toxin. It is not a purified, it has some media components also. So some people, they do the purification of a toxin first and then they do detoxification. There are two processes. We were also doing first the crude toxin. What we were doing is detoxifying it, and detoxification is done by addition of the formalin at a certain pH. So this formalin attaches to the lysine moiety of the toxin and it inactivates it, and this is called as a detoxification. So once you do a detoxification of a crude toxin, you have the chances of certain media components which are there, and because the formaldehyde, it has the tendency to form sort of a matrix sink. So the molecules become larger and you have a chances of having the media proteins also coming along with it. And once it is toxided, then you do a purification. So and purification is again done by ultrafiltration. So you know the molecular weight of your product, then you use such type of ultrafiltration cutoff, and once the ultrafiltration done, you have a purified toxide. And this purified toxide, there are various tests which happens over here. So you have a test at a seeding stage, you have a test once the toxin is produced, because once toxin is produced, you have to find out whether this toxin has that much virulence which is required, whether this much toxin has been produced or not. For example, you have done the fermentation, but the bacteria has not grown enough. So the toxicity level of the toxin will go down, and you measure the concentration of toxin by various methods, like the Lyme's flocculation. This is an antigen-antibody interaction, and you find out what is the concentration. So in the process, you also fix that if the concentration of the toxin in fermentation goes below this level, you will not use it for your product. It has to be discarded. But based on your experience, based on your validations, this type of incidences comes once you are standardizing a material. But once you standardize a process, then this type of incidences never happen. So now this purified toxide lot which is there, it goes into different testing parameters. Even we have to perform the reversion of toxicity. Reversion of toxicity is normally happens for toxin which are formaldehyde inactivated. When you expose the product at a high temperature, there is a tendency of reversion. Okay, so you have to validate your kinetics of inactivation before you make a commercial product. So each stage, I want to stress upon that each stage in the manufacturing is validated, and you perform your all processes within that validated procedure. You cannot deviate from this. Now this purified toxide which comes is now ready for formulation. So like we have made a diphtheria, we make tetanus. So if you want to make a DT product, this diphtheria at a particular concentration and tetanus at a particular concentration, along with the adjuvant, you formulate a product which is a DT vaccine. So there is no individual diphtheria vaccine, but there is a DT vaccine and there is a monovalent tetanus toxide. Okay. So once this is formulated, then on a final formulation you have the release test, and there is a battery of tests which is as per the WHO guidelines, as per the monographs. So those tests you have to perform before you release a product. And in case of vaccines, it is always linked to a release which is given by the National Control Lab, because only in vaccine manufacturing the product is released by the national control, and there is a concept of lot release. Like in pharmas are you make a product and you release in the market, your quality control and QA releases it. But in this case, it is to be used in campaign, it is to be used in mostly healthy individuals. WHO has put a rider that once you release a product from your internal QA, that product along with the samples and protocols has to go to the national control lab. And this national control lab, depending upon their sort of a consistency approach, if they are obtaining a product from a manufacturer like us for so many years, so they know the quality, and sometimes they release only on the scrutiny of the protocols, sometimes they do partial testing, and for majority of the manufacturers which are newer in the field, they do 100% testing as well as scrutiny. Any new licensed product which comes in picture, it goes to national control, and national control completely do the analysis, they do the inspections of the facilities, they come here for documentation review and then release the product. So this is one of the product for what we make like diphtheria, and for pertussis. Now pertussis is an example of a whole organism. So we use 4 strains of pertussis, and the selection of the strains is based on the, there was a trial which happened in 1951 that is called an MRC trial which happened in London, and it was observed that the children who were vaccinated, majority of the children, I would say more than 90% of the children, were having antibodies to agglutinogen type 1, 2 and 3. These agglutinogens are on the surface of the bacteria, and these agglutinogens are not expressed in all strains of bacteria. So what you have to do in order to have 3 agglutinogens in your formulation, you select various strains. So we have selected 4 strains, like one has 1 and 3, one has 1 and 2. So you have to pool those. So majority of the manufacturers have 2 strains, but we have 2 additional strains which have 1, 2, 3 in order to have a sufficient quantity of those agglutinogens. So this is again a simple isolation, inactivation and formulation of a product. And then again you have to find out that in what ratios you have to add various components, because you are using 4 strains. So you have to fix the composition of strain 1, strain 2, strain 3, strain 4. So we have 40%, 40%, 10% and 10%, and this is again I am saying not randomly, it is again proven in the clinical trial. Till this part of the presentation we have discussed about the empirical approach of formulation of a product or manufacturing of the vaccine, and the basic principle what I told was the isolation, inactivation and injection. But this approach is not always valid for all type of products. There are many organisms which are non-culturable, they cannot be cultured, and in certain cases the main antigenic determinants is not exposed on the surface. So if it is not exposed on the surface, you cannot have a protective antibodies against it, and you need to have very big facilities for handling those. These products are to be handled aseptically. There are different zonings or the classifications to handle each process, because throughout from opening of a culture till it is harvested and formulated, it is done in a grade where you have A, B, C and D, and these classifications are based on the quality of the air you have in your environment. And the investment in such type of creating such type of facilities is too big, and going ahead with certain facilities where you have to handle organisms which are in biosafety level 3 or higher biosafety level 2, still it's a big investment. And most important is that you know that there is a disease, there is an incidence. If you start developing a product using this way of called empirical way of manufacturing, it takes very longer time. Now the Menafrivac, the incidence, the manufacturing which Dr. Jadhav will tell, it started in 2002, and in I would say in 2012, after 10 years we got the product in pipeline. Almost 10 years. 2009. Yeah, 2009. So almost 7 to 8 years it took. But still it's a very big time when you know that each year there is an incidence which is happening, and there is a cycle when this Men A was appearing, and there was a cycle expected. If we would have delayed our product further, there would have another big incidence or big sort of a deaths because of the meningitis. Now this is basically the polysaccharide vaccine. The polysaccharides are originated from the capsule which has this called envelope capsule which is surrounding the bacteria, and these capsular vaccines have been found very effective even for hip, for pneumo as well as for meningitis, but in certain cases these capsular vaccines could not give perfect immunity. There were certain reasons why it could not, because they were not able to generate the T cell immunity which is called the T helper cell generations. So what is shown over here is, here it is a bacteria where this is a capsule. What you do, you take this capsule, ferment it, purify, and then you conjugate it with the carrier protein. Now this is a carrier protein which can be any, like we are using CRM 197 which is originated from diphtheria. It can be from diphtheria toxoid and from tetanus. Tetanus has been considered as a good carrier protein because it gives a good immunogen. Now what you are doing is, from a simple structure you are making a complex, you are giving a complexity to the antigen, and one of the definition of a component to be more antigenic is how much complex it is. So it has a diversification. So by just addition of a protein through a chemical linker, this molecule is generated which is a conjugated product. So it has tendency to give a lot of T helper cells and prolonged or long lasting memory for immune response. Now this, especially the hip polysaccharide vaccine has been found effective in adult population, but the children below 2 years of age, this vaccine was not found effective. So there the conjugated vaccine was found very effective, and now it is given from the child of age from 1 to even 47 years of age, it is recommended. Now the meningitis which is caused by the organism called Neisseria meningitis has 5 serotypes, which is A, B, C, W135 and Y. Now the vaccine is available for the A, C, W, Y components. For B also it is now available, but very recently it is available for B. But for B it was difficult to make. The reason was that for earlier product it was the capsule which was playing a major role, but for B it was not the capsule which was playing a role. So the scientists have to go for the other components which are over there. And why this capsule was not able to make the product? Because of some sort of a what you call a chemical moiety, which is a poly alpha 2B and acetylmuramic acid, which is common in the human system also. So the first definition of antigen or a vaccine is it should be not a human. So it becomes a sort of an auto antigen. So that's why the capsular vaccine against B didn't work. Lot of trials have been made, and this led to an approach that the empirical way of making a vaccine is not good enough. So scientists have to think in a different way which is non-empirical to make certain products, and this led to looking at the genome of a product. And here is the concept of reverse vaccinology, and the first product which has come up as a vaccine against meningitis B was developed by Novartis, and it is based on the reverse vaccinology. I'll highlight the bullet number 3 regarding the CMI, because we are also working on the whole cell pertussis vaccine, and we are developing an acellular pertussis component where we would like to have more sort of a response against TH1 and TH17. So we are adding one component which is developed by one of the companies in France called Gentacil. They have given us the adenylate cyclase. Adenylate cyclase is one of a bigger antigen which is found in which is a part of the outer membrane of Bordetella pertussis, and this all is being done to avoid the reoccurrence of pertussis. So this is an additional antigen which we will be adding. Now we have still to find out whether this will be treated as an additional antigen or we will treat it as an immunomodulator. So we are performing certain experiments. Manish is involved in that to see what type of cytokines are there and whether it will be a part of a formulation or it will be taken as a separate. So it is a sort of a pre-development work. Still we have not standardized the process, but this component, we think that this is the one which will make a shift from a whole B cell to a Th1 and Th17 response and avoid the recurrences of the product. Now these are the challenges which we have to now do. We have to face these challenges to make the product against all these infectious diseases, including bacteria, viruses, parasites, and there are certain vaccines against, develop the vaccines against where the therapy is there. So vaccine is normally for prophylaxis, and the vaccine as a therapeutic, the concept is also there, the question which you have asked. But still there is no very clear signal the way forward for making a vaccine against the cancer. People are working on it, but strictly speaking there is no such type of a specific distinguishing product which can only impact the cell which has a cancer impact and the normal cell. Yeah. So the earlier the vaccines were normally made for the pediatric population, considering pediatric population in picture, but now if you see, the vaccines are to be made for infants, children, adolescents, adults, elderly. So the target population is changing, and as the empirical approach is not able to give any sort of an output from for these answers to these all infectious diseases, so the scientific community is moving from the empirical approach to the other advanced approaches. Now, in order to have what they have done is for a meningococcal B vaccine which was made by Novartis, they have the full genome sequence of the organism, and once they have a full genome sequence of the organism, they try to in different strains, because Meningococcal B has got lot of variability in the strains. So what they have done, that they have isolated certain genes from, they have picked up around 600 genes which could induce express the proteins. Then from these proteins they have injected the animals, found that these proteins can produce the neutralizing antibodies which are bactericidal in vivo or in vitro. From there they have screened up to 325 from 600. First of all is 2000, then 600, then 325, and ultimately they could reach to almost 7, and out of the 7 they broadly selected 4 such type of genes which can express the proteins, and these are the OMPs or the fusion proteins. So this is called a reverse vaccinology. Now it is not from the organism as it is. What they have done, they have completely done the full genome sequence, and for this organism the genome sequence was made available in 2000. The first product for which genome sequence was available was a Hib. It was done in US, and this was the second. But then this genome sequence was utilized as a tool to develop a vaccine by reverse vaccinology. So this is the whole concept of reverse vaccinology, that you are not doing as an empirical, as a product, isolate this, but you are looking at the genome and trying to find out what are the epitopes or the antigenic determinants which are expressed by specific genes. So this, and in order to have the reverse sort of a vaccinology, you have 3 tools. One is a host genome, second is the pathogen genome, and third is the information on the proteomics and the bioinformatics. Now what I discussed here is the pathogen genome, which is this slide. I met him in London in one of the conferences where he presented his case study on Men-B, and this is the only successful product which is currently available. So by reverse vaccinology, what you have done is from the computation you have done in silico evaluation of the candidate genomes, and then taken out those, expressed those recombinant proteins and done the immunogenicity study. And once the immunogenicity in terms of its bactericidal activity is proven or neutralizing activity is proven, you have gone for the development. But see the time scale. The time scale here is 5 to 15 years or maybe more, or maybe a failure also. But here, as you understand the full genome of the organism, you are very sure that its virulence part is somewhere or the other part of the gene, which you have to hunt for. Now the tools which are available, they are so efficient that you can find out, but lot of efforts are required. So now the whole scientific community, especially for those products which are difficult to manufacture using empirical, they are moving towards this direction. Now there is a free website which is available on the web, where if you know the genome of your interest, if you know the whole genome of a product, of any organism, if you put that over there, you will get the complete answers to your questions related to these factors. Where is actually the adhesions are there, where are the transmembrane domain is there. So these, now this is available on the portal website, and we are exploring this website again with one of the students working at Serum for the Pertussis, because currently what we said is we never know the recurrence, but recurrence we do not know because we do not know the strains. So that study is not done anywhere. So we are trying to do that study. We will we are collaborating with CMC Vellore also. CMC Vellore has done under ICMR some of the what you call selection of serotypes. They have certain organisms, they have done the genome sequencing also. So we will collaborate with different hospitals for getting those serotypes, and with this tool we are trying to figure out what are the strains which are basically available and circulating, and their virulence, to further make a product which will avoid all the recurrences which happens in future. Now the second part is the first was the genome sequence from the pathogen. Now this is, now there are individuals who have got the infection, who have got the vaccination. So even after infections they are protected. So if they are protected, what is there in their genome which helps them this sort of a protection? So there what you do, you take out the B cells. Now the B cells are the one which are the plasma cells. They are the effector B cells, means activated B cells. So from those B cell population, you have to culture them and find out what type of IgG, what type of gene sequence which is there in their B cells, which is effective, helps them in protection. So again what you are doing, you are taking a case study from a human being which is effectively saved because of the vaccination or because of the pathogens. So now you are exploring another tool which is a human system. Okay. Again by the same way you will express those, you will find out the specific type of gene which is responsible for making those antibodies. In the next slide it will be a little more clear. Now here you have taken the B cells from the human which is vaccinated or protected. You do the cell proliferations, and do the IgG gene sequencing. Now this FAB, this is the portion which is antibody capturing. Now this FAB is generated through this. You have to find out whether this is protective or this is not protective by screening a lot of B cells which are there. And once you see in the system that it can capture a particular epitope, that means this epitope is now a good antigen which is mimicking the reflection of this B cells which has protected the individual. Now you have developed this antigen indirectly by these antibodies. Now this antigen can be safely used to human to generate again such type of B cells which will protect the individual for further infections. So again it is a reverse vaccinology. Okay. This looks very good in pictures, it looks but once people they work on it, and of course the rhinorapolis is a proven fact. So working on these aspects definitely lead to, we have not seen any product coming out of this, but there are so many products which are in development. With this technology, even working on one pathogen, you will have at least maybe 15 to 20 different candidates. So those 15 to 20 different candidates you have to take forward in the R&D. So you can again see the load on the R&D and of course the investment in the R&D. Now this genetic techniques, they are not only used as a reverse vaccinology, they are also used to develop a strain. For example, if you have a strain of influenza or HIV, even meninge, there is a lot of variability. HIV is the most variable. Now this is how one can develop a strain from, I want a desired strain which has the expression of H and N antigen. These are the heme agglutinates which are present on the surface of the influenza virus. So we have selected, for example, two strains of virus. From this I want strain, from strain 1 I know I want 2 genes, and from strain 2 I want 6 genes. So what you do the splicing and remove all those fractions which are virulent and take out those portion, and then they introduce into the plasmid and put into the circular DNA. Here also you take out the 6 and integrate in that circular DNA and cultivate it, and your new strain is ready. Now this new strain, why you do modification to the new strain? In order to cover most of the variability which is existing. So still it may not be fully effective, but if it is 70%, 80%, still it is better than 20%. We are also working on one of the product. Now this is a BCG vaccine. Now BCG is a very old vaccine, but if you go back and look at its efficacy, it is always a controversial product. Even in a Chingalput trial, the efficacy is said to be 0 to 60%, but this is the product which is currently available for vaccination, and you can imagine that there is a lot of deaths which are happening. One is death because of HIV, another is malaria, but the second death, most deaths which are happening is because of the TB globally. It is not that current TB vaccine is not protecting, it is protecting, but then efficacy varies from one to another. So we have an interaction mainly the infants. Yeah, mainly the infants. We are working with one of the company in Germany which is called VPM, and this is under Stephen Kaufman from the Max Planck. They have developed BCG which is a recombinant. What they did is they have taken the listeriolysin gene from the Listeria monocytosis. This listeriolysin gene is introduced into the, it is an extra chromosomal, it is not a part of the chromosome. So it is put over here, and simultaneously there is a gene called urease gene which is already existing in the bacteria, BCG. Now this urease gene, what it does, when it utilizes, it liberates ammonia, and when it liberates ammonia, the pH of the product, the pH of the system is 6.5, and this avoids the lysis of the lysosome. Okay. The moment you introduce Listeria monocytosis, Listeria gene, and remove the urease gene, the pH goes down below 6, and this Listeria monocytosis creates pores in the membrane, and because of this the antigens are expressed out. and once they are expressed out, they are processed through the phagosomes, and you can see that they are expressed by the dendritic cells to the immune cells, which leads to production of lot of IL-17, IF and gamma, which is a very good and perfect CMI. It's a cell mediated immunity, and cell mediated immunity is very important for an organism which is intracellular. So we are already now from this we are developing two type of product. One is a BCG, which is a pediatric vaccine. Another is there is a BCG in what you call concentrated BCG, which is also used for the bladder cancer. Current BCG also we are using for bladder cancer. So we are developing these two, and these two products are in clinical trial. And for bladder cancer, we have already done the phase 1 safety. The product is in phase 2 in Europe. Both these trials are in Europe. And for a pediatric vaccine, we are convening a trial in South Africa, where we are giving a product to the, I would say, HIV exposed mothers. Now, this bacteria has got one, the virulent bacteria has got one very this thing, that if your immune system goes down, if there is a lot of incidence happening of TB, is an indirect evidence of the HIV infection, because your immune system goes down and HIV, it is shown. So in those population, if this product is proven safe, it is a challenge. In India also, we are going to have a phase 2/3 or phase 3 clinical trial, again in the population where it is a recurrence which is happening. BCG has a recurrence. It doesn't that if you give the antibiotics or anti-TB drugs, it subsides, but there is a chance of recurrence. Now this recurrence is happening because of the same serotype or because of the different serotype, it still has to be evaluated. In those cases which have been given, there was a recurrence, which have been given antibiotics or anti-TB drugs, those after removal, clearances of the drug from the system, we are giving our product, just to find out whether the recurrence incidence, which is almost 12 to 13%, goes down to 2 to 3% or 4% or not. Again, it is an indirect evidence of avoiding recurrence and effectiveness of the product. Now, this is a story of the HIV vaccine, where so many products have been developed, but still you will see there is no effective product which has come up, and the last thing, which is this one, if you see this, these are all dead now. These all technologies to develop the product has gone dead, and the only one which is there is the only CMI. That for HIV vaccine, you have to develop those antigens or express those genes which can work on CMI, not the neutralizing antibodies. And there are few products which are in, I would say, phase 1 or phase 2 trial. Yeah, and this trial has two types of antigen. One is a GP120, and for GP120 basically for the neutralizing antibodies, and for the T cell immune response, what they have taken adenovirus as a vector, where they have incorporated surface antigen so that the memory against the B cell is increased. So it's a combination of the antigens which are incorporated into the adenovirus as well as the GP120 expressed protein, GP protein. GP120 is a surface protein which has been found to be in most of the strains, because what you do, you select the proteins which are available in majority of the strain. If you that is how these are taken out. So GP120 has been found in majority of the viruses, and that's why it is selected. But it was alone, it was a failure. In combination, in the sense you take another protein, still it was a failure. So it is an indication that neutralizing antibodies alone are not enough. So you have to have a CMI. So for CMI, what they have done, because viruses, they induce a lot of CMI, so they use adenovirus as one of the vectors and incorporated certain surface antigens, which you will have more details once you go through that reference. So it's a combination of a product which can induce B cell immunity as well as T cell immunity. But still you can see it is giving around 31%, but NIH of course has taken this product as a lead, and they are working on it. And this is the virus which has been shown as the maximum drift. it's spontaneous drift, means whatever you are injecting now, you will have different sort of a thing which is coming out. It's a lot of mutation. So one is a shift which is slow, and there is a term called drift which is very fast. Different scenario. Means you have the patients from dying from tuberculosis. There is lot of meningitis cases, and this meningitis cases happen due to one of the fungal infection, which is a cryptococcus. And cryptococcal infection in a patient means is an indication of HIV, because cryptococcus is one of the commensals. And in this case, what happens that the commensals, they overgrow your and they overthrow, I would say, the immune system, and they multiply. So there are incidences of cryptococcus, maybe around 70% of the HIV cases dying of meningitis is having cryptococcus. And what I told is the BCG is the tuberculosis. Now if you see the vaccines which are made till 30s, they are on the empirical approach, and then the vaccines from empirical approach we have gone up to a recombinant product up to 1980, and in 1990 the concept of the conjugation or glycolization happened, and till 2010 we worked on the reverse vaccinology. But now if you see on the other part, the concept of the synthetic biology based on the RNA techniques, then structural vaccinology, and addition of the adjuvants, specific adjuvants in the formulation. These are the concepts which are coming up to develop the newer products. So now in reverse vaccinology, if one genome can give you a data, then why not the whole genome? Now in a human system, there are more than, I would say, 25,000 genes which are there. So the concept is, why not explore all the genes? So this is called as a system biology. Now the system biology is, you have certain tools where you can know the complete genome sequencing, you can know about the RNA, you have the data on proteomics, you have the data on lipidomics. So when you combined all the information, and you from there you can develop a product. So this concept is there now. Instead of looking at one gene, where it is better to explore the full variety of the genomes which are there in the human body, to develop a product which is specifically required for making a vaccine. Again, this is by having the information, by shortening your gaps of knowledge, selecting those antigens by reverse vaccinology, selecting good delivery tools, and selecting a good adjuvants which can give you either B as well as T immune response. You make a rational design to make a product, and this is the ultimate, where you will have all tools available to identify the antigen, and you can design your formulation based on the parameters which are addressed over there.